So we'll be moving on now for the young scientist talks. So I'd like to acknowledge the support from UPAB for us to organize this meeting as well, and to to provide fellowships for the young scientists to participate in the meeting that will occur in October. So I'd like to ask Fernanda Leomil from Unifesp, Brazil, now to put her presentation on.
Can you see it?
Yes. So
hi,
Fernanda,
everyone.
you have 15 minutes. So when there is 5 minutes left, I will warn you, okay?
Okay, perfect. Yeah. So hi, I'm Fernanda Leomil. I'm a PhD student at Unifesp in Brazil, and a visiting PhD student at the Max Planck Institute of Colors and Interfaces in Germany. And I'd like to thank the organizer for setting up this nice event, and also for the opportunity to talk to you today about the stability of membranes containing different types of anionic lipids. So the cellular biomembranes, mainly composed of different phospholipids and incorporated proteins, act as a boundary for the cell, separating its intra and extracellular media and creating a delicate control of material exchange between these two. Besides that, it also plays an important role in the cell mechanical stability. Under some mechanical stimuli and even physiological activities, the bilayer can rupture through the opening of a pore that will tend to close, allowing the restoration of the membrane integrity. And this ability to reseal a pore defines whether a cell will survive after a stimuli that can cause the membrane to rupture. Although this barrier created by the cell membrane is very important for the cell stability, sometimes it may be interesting to temporarily allow the entrance of otherwise impervious substances. Particularly, electroporation is a technique that consists in the opening of transient pores in the membrane in response to the application of a high-intensity electric field. And it has been used for different medical purposes, such as for cancer treatment in combination with chemotherapy, for gene therapy and DNA vaccination. And although this is quite a well-established technique, still little is known about the mechanisms that are responsible for increasing the membrane permeability after electroporation, and also about the ability of the membrane to reseal after that. So model membranes have been widely used to study cell phenomena without the interference of adjacent phenomena that can interfere with each other. There are simplified versions of the complex membrane system, being composed of a minimal set of components, and also they allow the modulation of several parameters of interest, such as the composition. And they are still evolving, allowing them to get closer and closer to the reality of the cell. Among all the existing models, here giant unlamellar vesicles, or GUVs, are the most suitable, since their size allows to obtain direct information about the pore dynamics after electric pulse application under an optical microscope. The pore dynamics in a lipid bilayer can be described as a balance between the membrane surface tension that reflects a stress to which the membrane is subjected to and tends to open and stabilize the pore, and the pore edge tension that reflects the energy cost of maintaining an opened pore in this membrane and thus tends to close the pore. The stability of neutral POPC membranes has been already well elucidated. As we can see in this video, after the electric pulse is applied, pores are open and readily reseal. It's not playing. Is the video playing?
Not yet. Let's wait.
Yeah, I guess I have some technical issues.
Yeah, cannot play.
Yeah, so I guess that won't work. But the idea is that for POPC membranes, the pores are completely resealed after the application of the pulse. However, when we have POPG in the membrane, that is an anionic lipid, the bilayer completely disrupts through the completely indefinite open of the pore, leading to a phenomena that we call bursting. Also, in those cases, some UVs that apparently restored their integrity remain in a leaky state, revealing the persistence of submicroscopic pores even for some minutes after the end of the pulse. In our recent study with PG, we also showed that the fraction of compromised UVs after electroporation increases with the increase of the fraction of PG in the membrane, and that the origin of this destabilization is a twofold reduction in the porage tension for those UVs, as we can see here in this graphic, for UVs containing 50 mol% of POPG. So these results with POPG was an indicative that the surface charge may impact the membrane stability after electroporation. However, this PCPG model is still very basic when compared to all the complexity of the biological membranes. So it would be interesting to study the impact of anionic lipids of higher biological relevance on the membrane stability after electroporation. And that was the main goal of this work. So we explored 3 different anionic lipids. The first one was cardiolipin. It can be found in the inner mitochondrial membrane, and it plays multiple roles related to energy transformation, apoptosis, and membrane integrity. And cardiolipin is very different from the other phospholipids, since it has 4 acyl chains, 2 negative charges on its polar head, and a negative intrinsic curvature. We also explored PI and PIP2. They can be found in the inner monolayer of eukaryotic plasma membrane. They play important roles in the regulation of several cellular processes, including cell signaling and membrane transport. PI is quite similar to PG. It has 1 negative charge on its polar head and a neutral intrinsic curvature, while PIP2 is also very different. It has 3 negative charges, a high degree of unsaturation, the acyl chains, and a positive intrinsic curvature. So our UVs were prepared by electroformation for stock solutions containing POPC and increasing fractions of one of those anionic lipids. And here, this lipid mixture is spread on conductive glasses that are sandwiched with a spacer in the middle. And this kind of a chamber is filled with the inner solution of the UVs, which in the case is sucrose. And then this chamber is connected to a function generator that will accelerate the growth of the UVs. And this whole procedure takes from 30 minutes to 1 hour to be completed. Those UVs were diluted in isotonic glucose solution, so that when they were observed under phase contrast microscopy, due to the differences between the refractive index of the sucrose inside and the glucose outside, it was very easy to observe the membrane as well as any discontinuity in it, such as the one caused by the opening of a pore. For the electroporation experiments, these UVs were put in an electroporation chamber that has 2 parallel electrodes inside, separated by 500 micrometers, and these electroporation chamber were connected to a microporator. So initially, we sought to investigate how the inclusion of increasing fractions of different anionic lipids would affect the destabilization of the membrane upon electroporation. In order to do that, we observed a population of UVs for about 10 minutes after the application of the pulse, and we measured the number of UVs that underwent bursting, as we can see in blue. That is the complete disintegration of the membrane, and also measured the number of leakage UVs, that are vesicles that lose their optical contrast as a result of the mixing of the outer and inner solution. And since these 2 events are directly related to the integrity of the membrane, we combined them two in a fraction of disrupted UVs. So this figure shows the fraction of disrupted UVs as a function of the fraction of anionic lipids in the membrane. And the tendency here is very clear. We can see that the membranes becomes more unstable upon electroporation as we increase the fraction of anionic lipids in the membrane. As I mentioned before, some UVs remain in this leaky state upon electroporation, and that reveals that some submicroscopic pores are still open even for some minutes after the end of the pulse. So we decided to investigate the time dependence of this contrast loss for those leaked UVs. Can you hear me?
Yes,
Yes.
we can.
Yes, we can, yeah.
Yeah, for some reason my slide completely froze. So I'll try to, no. I'm sorry, I try to be, I try to be very fast. Okay. Can you see my slide or not yet?
Not yet.
Okay, then I share it again.
Yeah,
Can you see it now?
Yes.
Yeah.
Okay, I'm super sorry. But yeah, so this is the leaked UV and now the video is working. So this figure shows the relative optical contrast as a function of time for UVs of different compositions. So we have neutral UVs, UVs containing 30 mol% of PI, 10 mol% of Cl, and 50 mol% of PIP2. And we can see that for all cases, the contrast loss completes within 1 or 2 minutes. And what we also saw is that increasing the fraction of anionic lipids did not seem to alter the permeation rate. So as I mentioned before, the membrane stability upon electroporation directly depends on the pore resealing ability after the pulse. And this is directly related to the pore edge tension. So in order to measure pore edge tension, it's necessary to create a pore in the membrane and then measure its closure rate. That is the pore size throughout an entire video. And this closure rate can be correlated to the edge tension according to this equation here, where gamma is the pore edge tension, eta is the viscosity of the aqueous medium, T is times and C is a constant. And from the slope of this third stage, one can obtain the pore edge tension. Well, it's possible to track pore size manually along hundreds of images. However, it has shown to be time-consuming and also to generate inaccurate measurements. So in order to reduce analysis time and to generate more consistent and reproducible data, we have been developing a software called POET to automatically calculate pore edge tension in UVs. So POET was developed in Python. It's a free and open source software that works with TIFF image sequences obtained by phase contrast microscopy. And it has its own editing interface where the user can crop, rotate, or slice the video in time prior to the analysis. And only a few parameters have to be inserted by the users, such as the video acquisition rate, the resolution of the microscope, and the viscosity of the external medium. And the user can also choose from 2 different modes of analysis, the fast one or the complete one. And if the complete one is chosen, then in the end of the analysis, he will have access to a post-processed video containing all the radii that were measured throughout the entire video. So once the analysis is completed, a second window will pop up with the pore dynamics graphic containing the original data in red and the smoothed version in blue. And this smoothed version is used to generate a suggested linear region that will help the user to define the correct region for pore edge tension calculation. At the end of the analysis, the pore edge tension is directly shown in the main interface, and also the graph as well as all the parameters that were used for the analysis and the radii measured throughout the video are saved directly on the user's computer. So we asked 3 different analysts to manually measure the porage tension in a group of 6 UVs and compared the results with the ones obtained by POET, and the results were quite similar. And also the pore dynamics graphic were quite the same when a well-focused UV was considered. So as we can see in this figure here, where the same 3 analysts were asked to mark the extremities of a pore, a good match between the analyst and POET is expected for good or well-focused UV. However, for badly focused UVs, a large measurement variation within analysts and also with POET is expected. Well, but I would say that the best result of POET is that it allows a 96% reduction in the analysis time. So we used POET to calculate the porage tension in our UVs, and this graph here shows the porage tension as a function of the fraction of anionic lipids in the membrane. And we can see that similarly as we observed for PG, we see a 2-fold reduction in the porage tension for membranes containing any 50 mole% of any of those anionic lipids. So here we show that there is a notable membrane destabilization for UVs containing 50 mole% of anionic lipids, and that despite all the shape differences and intrinsic curvatures that we see for these lipids, apparently the determining factor for membrane destabilization upon electroporation is a minimum membrane surface charge that is capable of causing a reduction in the porage tension.
5 minutes, Fernanda.
This graph shows the edge tension, but now as a function of the surface charge. And here yellow is highlighted the surface charge that is known to cause the 2-fold reduction in the porage tension for membranes containing 50 mole% of PG. So as expected, since PI and PG have quite a similar area per lipid head group and charge, we also saw a reduction in the porage tension in the same surface charge for membranes containing 50 mole% of PI. As for PCl, although it has twice the charge, it also has almost twice the area per lipid head group as PG. So at the end, they have a similar ratio of charge per area, and that's why we only see a reduction in the porage tension for membranes containing 50 mole% of Cl. As for PIP2, indeed, we were expecting a reduction in the porage tension for lower molar fractions of PIP2, since it has 3 times more charge and the same area per lipid head group. However, apparently several factors can favor the desorption of PIP2 from the membrane, and it's apparently what was happening is that this result here for membranes containing 50 mole% of PIP2 was related to a lower amount of PIP2 in the membrane. And so in this work, we show that the membrane indeed plays an important role in membrane destabilization upon electroporation, and that this reduction in the membrane stability is related to the reduction in the porage tension, but also possibly to membrane asymmetry, which is something that I'm investigating now. And we also show that with POET, we can analyze in a more consistent and reproducible way a larger amount of data. So I would like to thank my group in Brazil, especially to my supervisor, Karin Hiske, as well as my group here in Germany, especially to my supervisor, Rimiana Dimova, and also Marcelo Zocoler. And I would like you to invite you again to check our recent paper in Advanced Science, as well as to register for this conference that, as was mentioned before, will be held online this year in October. And if you're curious to know what else we can do with giant unilaminar vesicles, also check this fantastic book. And that's it. Thank you all for the attention, and I'm sorry for the technical issues.
Thank you for the talk. We have one question now here. I will make for you. It's from Matia Morandi, and he asks about, have you tried testing the same system in physiological buffer conditions where presence of cations may screen negative charges?
Yes, we have. And actually, this result is published here in this paper, Too Close or to Collapse. Then yes, so calcium indeed prevents all these effects because of this, because it's like neutralizing all the charges. So yeah.
Okay. He also asks why to use PG rather than PS, which is more abundant in cells.
Yeah, I also have some work with PS, but we were using PG because the lab was already doing a lot of work with that, and it's relevant for bacteria. So yeah, there's not a specific issue, but I also have results with PS.
There's another question from Emerson Carmona. Thanks, nice work. I'm curious about the electroporation process you use to form the pores GUVs. Is the membrane sensing a transmembrane voltage difference when you apply the pulse? If that is the case, what will be the transmembrane voltage applied?
Exactly, yeah. So the pores to open, for the pores to open, it's necessary that the critical transmembrane potential transpasses a minimum value. And yeah, so the pulse, normally it's at 1 kilovolts or something like that. But that will depend for the size of the GUV as well as the parameters of the pulse.
There's another question from Agustin Mangiori. Congrats, it's a great work. Have you tested in any way what the amount of charged lipids that you wanted to include in the vesicles were actually incorporated?
No, this is a very good question. We actually, when we do our GUVs via electroformation, we are not sure about the real amount of charge in the GUVs, and also about how is the asymmetry or the symmetry of those GUVs. So this is something that we test later. But yeah, we can say that some preparation has more or less charge. For example, when we do zeta potentials with the lipid mixture, but we cannot say that it's exactly 10 or exactly 20, but we can say that the former sample had less charge than the second one.
There is one question now from the panelists, from Rafael. Beautiful talk, 2 short questions. When, where is the software available? And the other one is, what's your next step?
Yes, so this software will be available, I hope, very soon. And it will be open source, as we mentioned. So we will give the code, and it will be in GitHub. So yeah, as soon as it's out, I'll let everyone know. And about the next step, so as we mentioned it's possible that the membrane asymmetry of this asymmetry of charges in those membranes are also playing a role in the destabilization of the membrane, since it can generate spontaneous curvature, and this would help to destabilize the porated membrane. So now I'm investigating how asymmetric membranes behavior upon electroporation. So now I will have charges only in one of the leaflets, for example.
Okay, there are questions now. People, I think they're gonna open their microphone. Mauricio, would you like to make your question?
Oh, yeah. Thank you, Fernanda. Beautiful work and nice presentation. Thank you for participating so well. I wonder if you have considered the fact that several groups have shown that electroporation causes chemical change, oxidation of the lipids, and perhaps some of the difference in the lipids could be explained by this. I don't know if you have considered this fact.
Yes. So in this paper, we also consider that. But at first, we assume that this is not something that we should worry, because in principle, this would take a longer time to be observed. I mean, the oxidative effect of the lipids. But yeah, we consider that it's something that we have in mind, like all the time, that this can happen. Mostly for PEEP2, I would say, yeah.
Thanks, Fernanda. Before we move on, there was, there's another question from Leandro. Leandro.
Actually, I have one question, Fernanda. Very nice presentation. I saw you showed for us a graph from the charge density on the surface of the membrane, something around 0.1 or 0.2 coulombs per square meter or something like this. When you make some calculations, you see that using those numbers, it's almost that every lipid is with some charge there because they are ions and so on. But can you do you calculate in your software the ionization coefficient of this system? I mean,
No.
that alpha parameter, because I think you can retrieve that number and compare with some other theoretical and practical works in the literature, only to double check your result, because those are known from PG and from PI.
Yes, this is a good idea. Thank you, because we
No,
didn't check that.
Yeah, I think it should be a nice idea.
Nice. Thank you very much.
Congratulations again for your team.
Thank you.
So, like, if there are no further
I think
questions.
Rosangela has one question.
Oh, so
Eduardo.
Rosangela, please.
Sorry, I don't know if you still have time, but first of all, Fernanda, thank you very much for your very nice presentation. It's very nice work. Really, congratulations. I have a very quick question. Is regarding your pore tension software. It's really curious. How big it must be the pore to be detected? Do you have a limited?
So that depends a lot on the acquisition rate that you are using, right? So if you use like a fast camera that gets to, I don't know, 20,000 frames per second of acquisition, then you can really define the pore dynamics with, I don't know, but I would say a few micrometers. Sometimes you have really high, huge pores, for example, 10 micrometers, 20 micrometers, and this relates to the most well-defined pore dynamics. But you can analyze like 5 micrometers or 10 micrometers also.
Thank you very much. We wondered to see your
Thank you.
software. Thank you.